So as you can see in this movie, I'm very interested in self-assembly in general, but we mostly work on proteins. This was actually quite tricky to put together because it was filmed with a drone on a very windy day, and getting the image to be stable with post-processing took several days. Anyway, so okay. So far, I mean, today we've heard a lot about proteins already, but yesterday and the day before we heard about prebiotic, perhaps what I would like to call soup, because in France, often when you eat soup, it's mixed, so it's kind of a homogeneous mixture with lots of small things, and the way I see then the biotic, what I would like to call ramen, because then you have like these bits and pieces of or macromolecules that we are trying to understand the properties of. And so I think so. So to put a when I was first invited to give to participate in this meeting, because I don't formally work on the emergence of life, I asked Liam, so why would you like me to come? And he explained to me that, well, protein assembly is important for the emergence of life. And actually, when I think about it, I think this is totally right. And I hope that through the principles that I'm going to present, we might be able to better understand this biotic ramen, or the properties of this ramen, so to understand how the pieces might have come together to make up the beautiful machineries that we see today, and that you can actually see in this picture. So now this is not a scientific picture. It's more of an illustration, an imaginary illustration based on known structures from Gail McGill. And one of the properties that you can already grasp here is that most of the proteins are not present in isolation. They are present as part of assemblies. So you have microtubules, actin filaments, lipid membranes, nuclear pore complex, and many other complexes, many of which, if you see here, are symmetric. Now, so in my talk, I would like to discuss mainly three properties that we've been working on that I think are important to keep in mind when you think about this biotic ramen. So the first one is that, as you will see, I will show you why we think, or why I think, that proteins are very sticky objects, meaning that the protein interactions are highly evolvable. The second is that symmetry among proteins is widespread, meaning that when new interactions form, it's often to form symmetric complexes. And finally, we will see how symmetry is a key property to give rise to higher order structures or mesoscale assemblies. So the basis for protein folding and protein interactions is hydrophobicity, amino acid hydrophobicity. So amino acids exhibit a wide range of hydrophobicities, which have been measured decades ago by many different people using various experimental methods. But the basis of most of these methods is very simple. It consists of measuring the free energy of transfer of an amino acid from a hydrophilic phase to a hydrophobic phase. So if you take aspartic acid, it's hydrophilic, it lacks water. And so most of it will stay in water. And so from the equilibrium, you can infer the free energy of transfer just based on the equilibrium constant. And so in this case, for example, if you have 6 molecules in water and 1 in the hydrophobic phase, or what represents the hydrophobic phase, so you just the free energy of transfer will be proportional to log of 1/6, so minus 0.77. If you now look at isoleucine, you will have the opposite. Most of the molecules will like to be in the hydrophobic phase, and now you have a free energy of transfer that's proportional to log of 6/1, so plus 0.77. So this is how you can experimentally measure hydrophobicity, but the way often bioinformaticians do is to actually measure amino acid propensities, which is strictly equivalent, except that instead of measuring the equilibrium based on an experiment, you measure the equilibrium frequencies. And so in different phases, which in this case are different protein regions. So if you take a protein and you decompose it into here 3 regions, the interior, the surface, and the interface. So the interior is buried, it's not visible, it's not accessible to the solvent. The surface is in contact with the solvent, and the interface is in contact with other amino acids with another protein, another protein surface. So what you can do is look at the frequency of amino acids in these different regions and derive a propensity scale, which I like to call a stickiness scale. If you compare the interface, meaning the interaction surfaces, relative to the surface. So you can look at the isoleucine frequency at the interface versus the surface, and if you see that it composes 6% of the surface and 1%, 6% of the interface and 1% of the surface, you will say that isoleucine has a stickiness that is log of 6/1, so plus 0.77. And you can do this for all 20 amino acids and derive a stickiness scale, which resembles greatly the hydrophobic scale, but also shows interesting differences. For example, that lysine and arginine are fairly different. So in the hydrophobicity scale, arginine is the most hydrophilic amino acid, meaning that it should be to the leftmost part in this bar graph. But you can see that arginine here is kind of in the center. And indeed, arginine is a great amino acid to mediate interactions with other amino acids. And this is why it's not so hydrophilic. It's not so, it is actually a bit sticky, much more sticky than lysine. Okay, so now with this scale at hand, what you can do is a simple exercise, which consists of taking a protein structure. Here you can see it's a homodimer, and then decompose this homodimer into the same parts that I mentioned before, the interior, the surface in light brown, and the interface that is in green. And then what you can do is create imaginary patches of amino acids. And for each patch that you roll over, measure the average stickiness of the patch. And you can do this for surface patches, and then you get a number of values of stickiness values. You get a density distribution for those stickiness values. And this tells you what's the average stickiness of a protein surface. And then you can do the exact same thing, but for the interface. And now you get another distribution of surface stickiness for interface patches. And then the question is, how different are these two distributions from each other in terms of number of mutations? So how many mutations on average do you need to introduce to a surface patch so that compositionally it looks like an interface? Anybody wants to make a guess? It's published, so you might already know. So, 3? Okay, so I have 3 here. Who says less than 3? Do I have 2? 1, 2. Do I have a 1 somewhere? Okay, so it's actually 2. So you only need 2 mutations, which is actually not much to have perfectly overlapping distributions. Which tells you that on average, whenever you have a mutation at the surface from a hydrophilic to a hydrophobic amino acid, you're quite likely to create what looks like an interface. It doesn't tell you what it's going to interact with, but it tells you that it has the right chemical composition to interact with another protein surface. So if you think about it, actually that makes a lot of sense, because if proteins were not sticky, Max Perutz and John Kendrew, whom you can see in the picture, would never have gotten a Nobel Prize, because X-ray crystallography would not work. Because X-ray crystallography relies on having protein crystals. And for a protein to assemble and form a crystal, it needs to be somewhat sticky to make contacts and form a lattice. Now, obviously, crystallographers spend a lot of time finding the exact conditions that will allow the protein to form a crystal, but nevertheless, it shows that there is some intrinsic complementarity that exists there. Now, if you look at the PDB and all the proteins that you can find there, this is hemoglobin, you can see the 2 axes of symmetry. One of them is actually pseudosymmetry. And if you survey the PDB, you'll see many complexes, perhaps not most, but like almost half of the complexes are actually symmetric, meaning that they form homolygomeric assemblies. And so you can, this is just a fun movie that also took couple of days to create. But there's actually one big issue with X-ray crystallography is that because you have a crystal lattice, you don't know in this lattice which contacts are physiological and which one results from the crystal formation. And so Susharita in my lab, when she joined, she worked on a methodology that will allow us to accurately identify those physiological contacts. And based on this data set, we could actually say that 30 to 50% of the proteins in the PDB actually form homoligomers, meaning that homoligomers are very frequent. The rest of them would be monomers. Doesn't mean that they are monomers in the cells. They might interact with something as well. But at least in terms of the protein, which could be a monomer, a homodimer, homotrimer, and so on. So 30 to 50% of them would be so. Now, this is actually not surprising if you consider many works that have been published over the years that explain why the self-recognition is actually highly energetically favorable when compared to interaction between different surfaces. We could discuss this for a much longer time tonight if you like. Now, what I just told you is based on X-ray crystallography data, and you might argue that, oh, but to get a crystal, it's easier to get a crystal when the protein is already symmetric. So maybe this is actually not a universal truth, and this is only true of X-ray crystallography data. So how about non-crystallized proteins? And that's where AlphaFold actually comes in. So when it was released, we started using it to predict homoligomers. I cannot actually show you the benchmark that we made, but I can tell you that AlphaFold is actually more reliable than I would like to say X-ray crystallography to predict homoligomers, because X-ray crystallography does not tell you if a protein is a homoligomer. It just tells you that the structure of a lattice, and you have to guess which one is which contact is real and which contact is not. And based on the set of proteins for which we knew which contacts are real and which contacts are not, we estimated that AlphaFold is more than 95% accurate in its prediction of homoligomers. And so that led us to consider 4 organisms: human, yeast, E. coli, and Pyrococcus furiosus. So we wanted to cover all kingdoms of life, and then predict homoligomers across their proteomes. So we ended up with a data set of 8,000 reliably, what we consider to be reliable homoligomers across these proteomes. And these are just a few structures that we predicted. Now, what was very striking to me in this prediction is that for many structures, for many homoligomers, we could not find a homolog that existed in the PDB, meaning that if you look in the PDB for such a dimer interface, you cannot find one. And the same goes for this one, which means that AlphaFold actually had predicted those oligomers de novo, or at least those interfaces, because this protein, you have a lot of monomers, monomer structures, but not a single homodimer structure. And these 2 homodimers are conserved from archaea to eukaryotes, from Pyrococcus furiosus. In Pyrococcus furiosus and in human, we see the same homodimer. And the same goes for the other one. Now, to convince ourselves that this was correct, we actually solved the structure of this homodimer. Martin did so. And the TM score, which indicates the agreement, is 0.96, which means that it's almost identical. And the same goes for this one. It was too small. Martin used X-ray crystallography. And the TM score here is 0.98. It's basically the same thing. You can see them superimposed. So we have good reasons to think, based on this and several other benchmarks we carried out, that these structures are actually, for the most part, largely correct. And so that actually also illustrates that AlphaFold, I think, opens up a lot of avenues for ancestral proteome reconstruction, which I think could be very cool. Now, what we could now do is actually take those predictions, these proteome-wide predictions, and project them onto bags of proteins that are known to form a complex, and now ask whether those complexes, if they are rich in homodimers or if they include homodimers, and we could predict that those complexes are themselves symmetric. And then, so we didn't have archaea here because there's no complex information for archaea, but for E. coli, if you take all the complexes known in E. coli, I need to hurry up. Oh, well. And you project this information, we ended up identifying that more than 90% of the actual complexes are homoligomers, and more than 95% of even the heterocomplexes are mostly symmetric, meaning that if you have an A-B complex, it's going to exist in the cell as A2B2. So if you imagine yourself in the cytoplasm of E. coli, that means that you'll see around you mostly symmetric assemblies. In eukaryotes, this number seems to be lower, but it's still a majority of the complexes that are symmetric. Okay, now we reach the second part of the talk, which the last part of the talk, so what does symmetry do? So symmetry creates multivalence, and a multivalence promotes higher order assembly. So this is something that we showed in a piece of work that so it's summarized in one slide, but for Hector that was 5 years of hard work. So in this work, what he did is he took a number of symmetric proteins from E. coli, and then tagged them with a yellow fluorescent protein so that he could visualize their expression in yeast. So he was taking E. coli protein, expressing them in yeast. So you can see yeast cells expressing one E. coli protein fused to YFP. And then he introduced point mutations at the surface of the protein. One point mutation, which by symmetry is repeated several times. So here you have a homo-octamer. So one point mutation means 8 mutations on the complex. And in this case, it's just single point, so glutamic acid to tyrosine mutation. And the idea is that the new sticky patch that we form by repetition will trigger the stacking of a copy of the complex with itself, and by symmetry, this stacking will occur both at the top and at the bottom and result in the formation of a filament. And so the nice thing with these filaments is that we thought we might see them easily in yeast. And indeed, when he expressed this mutant in yeast cells, now he saw a lot of these filaments. So we could know that this point mutation actually completely changed the phenotype from this to that. And that really shows that one point mutation can actually have a huge impact on the self-assembly of a protein, especially if it is symmetric. Now, later on, we even had another part which for one of the proteins, it was so prone to self-assemble that we didn't even have to mutate a charged amino acid to a hydrophobic one. We just had to remove the protection of a charged amino acid, meaning that here the mutation of this glutamic acid, that's what? No, that's glutamic. Oh, I'm completely, that's glutamic acid to alanine. So you could mutate it to anything, to a glycine, to an alanine, to even a threonine or a serine, would basically trigger the assembly. So you just had to remove the protective charge to trigger the assembly. So that was actually quite striking. So that's just a movie showing that one of these mutants expressed in yeast cells, you can see that, well, it just assembles. And it's actually amazing to see how the cell machinery handles those filaments, and also, I should say, those bundles of filaments. They don't seem to actually be unhappy about having this, that's another part of the story that we are actually still studying. Okay, so we also, so symmetry creates multivalence, and multivalence enables mesoscale assembly. And we are also analyzing this using a synthetic biology using the system that is illustrated here. So the system consists of a dimer and a tetramer. So you have a dimer and a tetramer, and the blue part, the blue domain, interacts with the orange one to enable the formation of this mesh-like structure. So when we co-express the dimer and the tetramer in yeast cells, what we see are these puncta that are formed and that are basically nucleating in new cells and just forming these what's so-called condensates. Now, the nice thing about this system is that it enables us to really, so a nice property that I don't have time to explain, that we can actually measure the phase diagram of the system in cells and therefore understand by then modulating parameters of the system, for example, the multivalence, see how the multivalence changes the phase diagram, or how the affinity changes the phase diagram, or how the length of the linker changes the phase diagram. So it's a highly tunable system for which you can actually have a readout of the self-assembly in vivo. Another important thing is that this system is actually also influenced by the parameters of the environment. And if you, when we were mentioning earlier today, this freeze, this dry-wet cycles, or this, that could actually be one way where you could have condensation, solubilizations, condensation, solubilization. And so many other parameters can influence the formation of these condensates, both in terms of the environment and in terms of the molecular features. Also the molecular features, if you think of dry and wet, that could also be the environment. And another thing that we measured to close with the beginning, another thing that we observed is that once you form a condensate, a protein-based condensate that is in a solvated environment, the recruitment of proteins within, the partitioning of proteins within those condensates is actually driven by stickiness, mostly. Meaning that a sticky protein will actually be naturally recruited or will naturally partition into condensates, whereas a non-sticky protein will remain outside. Yeah, so to summarize, so in this biotic ramen, I think we can assume that the proteins were quite sticky, and in that sense that they were likely to, or it was easy to evolve new interactions, that many of these macromolecules and proteins were symmetric, and that it was also probably thereby easy to evolve larger mesoscale structures and compartments. Now, how does this enable life? I still don't know, but I think we can think about it more. With that, I'd like to thank you and all my amazing groups that did this work, the funding, and yeah, thank you for your attention.